So what's happening Wednesday next week? Second midterm. What does that mean about Monday's lecture? Review. Exactly. Good. You guys are learning. This is wonderful. So this is the last set of new material that will be on the midterm. So after the midterm, we'll discuss what will happen the following week, which is different than usually what's happening. So as a nice review on our retroviruses, we get to do clicker questions to start out with today. LTRs in retroviruses are generated by the positive strand strong stop, the negative strand strong stop, blah, 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 the removal of the tRNA primer by RNase H, frame shifting, or the reverse transcriptase. 15 seconds, time to start guessing. 9, 8, 7, 6, 5, 4, 3, 2, 1. Liftoff. A number of differences of opinion here. We didn't talk a lot about positive strand and negative strand strong stops, but this is where you get to the end of the genome, either in the negative strand or in the positive strand. What's the first one that's made? First of the strong stops? Negative strand, because you have your template, which is the genome. And so that's the first one. That replicates out until what? The end of the repeat sequence, which is present at one end of the genome. Then that end, the positive strand gets degraded, and then you jump over to the opposite end, which is part of your LTR, but you actually haven't made the LTR, because what enzyme makes all of the nucleic acid is the reverse transcriptase down here at the bottom. So how you end up with that sequence has to do with all of these other things. How you make the reverse transcriptase is frame shifting, but where's the reverse transcriptase in virions? It's packaged in virions, so you don't need to make it, even though it's a positive strand RNA, and at least in theory, that could be something that you could use to translate and make all of your proteins. So it is the reverse transcriptase which is generating or making the LTRs. Yeah? Yeah, so what's making the terminal repeats? What's generating them? The fact that you have all of these recombinations is what happens then end up. It really depends on how the question is written in terms of that. And I, in fact, changed it this morning because I thought it was less clear with one of the answers that I had. So, okay, so our next clicker question for today is how many template switches are absolutely required for retrovirus replication? None, 1, 2, 4, 6.
Yeah, it says it right here. The first strand transfer.
I think in this case it doesn't really make a difference.
The measure.
5, 4, 3, 2, 1. So let's show the answers. Let's see. Good. Most people think 2. Yes, I agree with you. Those 2 are, again, once you've made the negative strand strong stop, what happens? You jump around to the opposite end of the genome, and that way you'll make 1 of your LTRs. And then your positive strand strong stop, that's before you have to go and make that second one of the LTRs. Why do you need 2 LTRs? For integration. So you have to have those sequences at either end that the integrase is going to bind to. So you have to have 2 LTRs in order to get this retrovirus replication to take place. So C is our correct answer. And our last clicker question for today is HIV-1 is mostly different from other retroviruses in its entry process, replication, mRNA formation, integration, or budding. And getting back to your previous questions about genome replication in terms of B. So, genome replication.
Integration or replication?
No, you're right. Integration is where the silver bond or silver. They are.
20.
As opposed to, in contrast, HIV is able to transport into the nucleus via the nucleophores.
10.
So this one integrates.
3, 2, 1. Okay, we've got even more of a difference of opinion on this one. And unfortunately, a difference of opinion with me as well. So, turns out you guys don't know this, but I actually get to see what the first answers are here. The first couple of answers were right, but people changed their minds, unfortunately. So, mostly different from other retroviruses. How? Entry process for HIV is what? Well, entry process into the cells. When we talk about entry, this is entry into the cell. So this is membrane fusion. Is that any different from other retroviruses? No. Replication. How's it replicating? So replication is, in terms of genome replication, what's different between HIV and the other ones? TAT regulation, but that's making, if anything, I think more of your messenger RNAs versus your whole genome. But the replication of the genome is made by cellular RNA polymerase transcribing from one end to the other. So that, to me, is pretty similar between those retroviruses. Messenger RNA formation. What's happening in HIV in terms of messenger RNAs relative to most of the other retroviruses? Okay, TAT's certainly involved, but what else? There's another difference that's not on here. So there's a whole bunch more proteins in HIV than any of the other retroviruses, at least the ones that we've talked about. So how do you generate all those other proteins? By lots of splicing, which has to do with mRNA formation. Integration is identical. Budding, pretty much identical. There's some slight changes in terms of how all these things are put together in terms of the final maturation process. So go with your gut feeling. Again, the first people, you don't know that you're the first people who voted, but stick with that, I think, will help. So the answer here is C. Okay, those people who need to take off, sayonara, have a good weekend. And are there more questions about retroviruses while people are moving here? Yeah?
There is, I believe, the 5' unique sequence on the 3' end of the genome when you replicate it. How is that degraded? Like, is it just because it's not double-stranded, or is there a process that?
Okay, so the question here has to do with the structure of the provirus versus the virus genome, which actually gets packaged. And that is that you have the 5' unique sequence also at the 3' end of your provirus. How is that gotten rid of? We didn't really talk about this, but when you think about the replication of the genome, that's all cellular polymerase. And so that cellular DNA-dependent RNA polymerase comes through, and there's a tailing site right after the repeat sequence, which is between the unique sequence at 3 and unique sequence at 5. Now, the question is, why do you not get tailing at the 5' end of the genome? And we didn't talk about that, but there's some very specific reasons that you don't end up with tailing that happens right there. So it's where the poly A tail is made by the cellular RNA polymerase. And so it's that messenger RNA, the positive strand, which is then getting packaged. So in the provirus, you've got your LTR, which has unique sequences from 5 and unique sequences at 3 at both ends. But when you've done that replication process, you lose the unique 3 because the start position for your transcription is after the unique 3 sequences. And then the poly A tail site is before you hit the 5' sequences at the other end of the genome. Does that kind of make sense?
With respect to the second figure question,
Yeah.
can you identify specifically what you mean when you say template switch?
So template switch, and so that's what, okay, generally what people are talking about. I should have brought up the same figure. So when you look at the replication process of the genome, so when you've replicated to one end, so if you think about it again, then we'll talk about negative and positive strand strong stop. So the negative strand strong stop is when you start from the tRNA primer, which is quite close to the 5' end of the genome, you replicate out from there. Then you're at the end, so it needs a template. So that then switches to the 3' end. And so that could be 3' end of the first genome that you started with, or potentially the second copy, which is inside the virion. So that's your first switch. The second switch comes when you've now made your positive strand. So first you continue that negative strand all the way out to the other end of the genome. And then you use the PPT, the polypurine tract, as a primer for the positive strand. That then gets out to the tRNA, and then the sequence in the tRNA which blocks continued replication until RNase H chews up that tRNA. Now you've got a copy of your primer binding site on the positive strand, which then loops around either in the same template or, again, to the opposite template, and then will copy from there to give you the rest of the positive strand of the genome. So you end up with double-stranded DNA. So that's the second of those template switches. You don't look completely convinced, but...
I think I'm just disagreeing with terminology is all.
Oh, okay, in terms of the template switching. That is really standard terminology, so it's not just me.
No, it's not an interpretation.
Okay, other questions, comments, worries? Yes?
And all this is done only by reverse transcriptase?
Yes, it's all the reverse transcriptase which is doing this. So there's no cellular proteins that are involved. The only cellular thing which is involved is the tRNA, which was packaged in the virion when you had the virion being released. Okay, other questions? So let's, again, switch gears once more. All kinds of fascinating things that viruses are doing here. Talk about tumor viruses. So these are, again, there's quotes for tumor viruses, and anyone in my molecular class says, who puts quotes on it doesn't really mean it. It is a tumor virus, or say the polyomaviruses, particularly the SV40 virus, is a tumor virus, but only under really extreme conditions. And we'll talk about what those extreme conditions are a little bit later on. In terms of the rest of the polyomaviruses, we've already talked quite a bit about the structure, where you've got these pentamers where hexamers should be in terms of the structure, so we'll talk more about that. The big difference now in terms of these viruses relative to everything else we've talked about so far is that they basically look almost exactly like eukaryotic chromatin. They're packaging their DNA in normal cellular histones, and it turns out that they're really wonderful models for studying eukaryotic transcription and translation because they are basically eukaryotic chromatin, only, as true for all of these tools, you have very large amounts of them, and they're really homogeneous, as opposed to trying to look at the whole genome. So that's a big picture, and partly why the reason that so many people have been interested in these polyomaviruses, and particularly SV40, which we'll be talking about now. The big message here is the so-called T-antigen, tumor antigen. Again, in the rare cases we could actually get tumors to be formed by these viruses. And the T-antigen, and that should really be T-antigens because there are multiple T-antigens, are really the key to how these viruses replicate. And in those few cases where they are causing uncontrolled growth, it's all about the T-antigens. And so a lot of what we know about tumor biology comes from studying these proteins from these viruses. So T-antigen, a lot of also just general cell biology. In terms of how they're replicating, they've got divergent promoters, which should sound really familiar based on what other virus did we talk about way back when? Decades and decades and decades ago, like before the last midterm? What else has right and left promoters? PR, PL? Sounds familiar. Good. Yes, it should sound familiar. Lambda. So Lambda has these divergent promoters as well. And alternative splicing should sound familiar because that's what we just heard about in HIV that obviously I didn't explain well enough to get that as far as a clear question. So again, we'll talk about where these guys come from, structure, binding, and entry. Of course, the important and interesting things are those that are here in red. And if anything, I could probably put this one in purple, the replication, because replication of this virus is really not that different than cellular replication, but almost everything we know about cellular replication was based on replication in these viruses. So it was first studied in these viruses before that. And then the transcription is similar because a lot of what we know about transcriptional regulation in cellular systems comes from the study of these viruses, and it turns out it's extremely similar. So I didn't bring my DNA tumor virus book. It's just about as thick as the RNA tumor virus book. But it's interesting because these viruses were found to be tumor viruses well after the RNA tumor viruses were found to be these tumor viruses. And so this was a bit of a surprise, in fact, that people had found these DNA tumor viruses, although it makes sense now from what we know about cancers as being a genetic disease, and these are our DNA controlling those diseases. SV40, which is basically when people talk about polyomaviruses, it's almost always going to be SV40. There's some differences, but for the most part, we're going to be talking about SV40. Simian virus 40 was originally in the Salk polio vaccine as a contaminant because the polio vaccine was being grown on monkey cells. Those monkey cells were infected by SV40. Later on, they got rid of that, and so it's not been an issue. But the original polio vaccine had SV40 in it, and that means that pretty much everyone in the US before 1960, and I'm not sure if there's anybody in here who was actually in the US before 1960, but they were exposed to SV40, and then there are antigens. If you take huge amounts of SV40 and inject it into rats, you will get lots of tumors. and so that's where the polyoma comes from. But you've got to put huge amounts in there, and we'll see why that is a little bit later on. But again, most importantly is that these viruses are really wonderful tools for understanding replication and transcription. They're not a really good tool for understanding structure because, if you remember from last time, it looks as if this should be a normal T equals 7 icosahedrally symmetric particle, but they're all pentamers. It's a little bit hard to see in here, but each of these have 5 sides, and these should really be 6 sides between a 5-fold axis of symmetry, which you have up here. There's the central pentamer with 5 around it. Here's your next pentamer with 5 around it. Should be 1, 2, 1. So in a T equals 7 lattice, but it's not quasi-equivalent because instead of having 6 of your capsid protein here, you have 5. But otherwise, it's basically a pretty standard structure. Just tells us that the structures of these individual capsid proteins, which are all exactly the same, are very, very flexible. So we talked about this before. Just the VP1s is exactly the same slide that we had before. 360 of these, you divide by 6, you end up with a T number of 6. You can't get a T number of 6 using that quadratic relationship, so it tells you that some of these things are very different. And again, it's because each of these VP1 proteins, which is the capsid protein here, assume different structures, whether they're a 5-fold axis or one of these hexameric holes, and each of those is a slightly different structure relative to each other. So it's an extreme flexibility in one protein, one amino acid sequence, but here 6 different potential structures that they can have, which is really pretty amazing. At least I think it is. They also have these beta barrel structures, which is very, very common for the virus capsid proteins. We talked about this for polio. We talked about it for the STIV, etc. So the actual structure here, particularly in the middle of it, is very standard, but it can assume all of these different other structures in order to make this icosahedral particle. So that we talked about before. What we haven't talked about is really the genome packaging. And the take-home message for genome packaging for all of these polyomaviruses, and for the papillomavirus, we'll be talking about later as well, is that it's completely normal. H2A, H2B, H3, H4, the normal histones, which are packaging nucleosomes inside the cell. So these are double-stranded circular DNA genomes. Not surprisingly, they bind to histones. They're replicating in the nucleus. When you do replication of double-stranded DNA, it's not surprising that you're going to be picking up these histones. And so this structure is extremely similar to, but much more simple than what you'd have in a normal structure. The other thing to look at here is just the process. These are naked viruses. There's no envelope involved. VP1, which are all of these pentamers, and then there's a VP2 protein just sitting underneath them. But 2 structural proteins, VP1 and VP2, and then all cellular proteins for everything else. So really very simple in terms of their structures. How do they get inside the cell? Sialic acid binding should be kind of a broken record now that sialic acid binding is pretty standard. What's a little different is instead of using normal receptor-mediated endocytosis, which goes into endosomes, these use caveolae, which is just another mechanism that cells use to pick up things from the outside environment. Caveolin is the protein that's involved there. And it turns out that these naked capsids are transported as capsids, so still apparently protein-coated, all the way to the nucleus, and the whole capsid is imported into the nucleus, and it's only degraded in the nucleus. So that structure only comes apart in the nucleus. So what's happening in the cytoplasm is that you have these virions being transported across. Yeah?
So on the last slide, for the VP2, why is it VP2/VP3?
So in some of the viruses, you've got VP3. Other viruses, you just have a VP2. Pardon?
It's neither or not, some have both.
I think a few have both. I'd have to check. Again, I think we'll see when we look at the genome sequence in just a second whether SV40 does or not. Again, I'll have to get back and check on that. But I think that some have two and some just have one. Yeah?
So you're saying the genome packaging is completely normal, but normal relative to
Cellular packaging. Now, there is one difference that you might notice here. What's missing from this? H1. And so H1 doesn't seem to be involved in the histones that you have in SV40 or any of these polyomaviruses. But otherwise, the core histones are identical. Okay, so here's our VP2 and VP3. So in SV40, which is what this genome is, we've got VP2 and VP3. But the main capsid protein, again, is this VP1 protein. Again, these are divergent promoters, basically just two promoters in the whole genome. Instead of being right and left, now they're early and late. But that, again, should look really familiar relative to what we've seen already in bacteriophage lambda. Also a circular genome. This one, of course, however, is much, much smaller than that circular genome that you have in lambda. It's only 5,000 as opposed to 50,000 in terms of the size. This intergenic region, not coding for protein, of course, is where you're getting all of your regulatory activity going on. Again, a lot like what you have in the case of lambda between PL and PR. You have, of course, promoters. You have to have terminators of transcription. What's interesting is that the termination of transcription in all polyomaviruses, in this case particularly SV40, they overlap with each other. So you end up with double-stranded RNA that's being generated here at the end if you have the early transcripts coming from the P early promoter and the late transcripts. What that seems to do is lead to induction of the RNA interference machinery and then degrades those RNAs. And so particularly it's important for degradation of these early RNAs, that when you get that double-stranded RNA formation. Lots of splicing that takes place both in the early transcripts and in the late transcripts, and it's that splicing which generates the different proteins. Of course, what's interesting in terms of regulation is all this intergenic region up at the top here contains a number of binding sites for both cellular proteins and viral proteins, and that viral protein in particular is the large T-antigen, which is going to come up again and again and again. Again, this is the tumor antigen, which they find in large quantities in these cells. This binds to a number of specific DNA sequences that overlap with TATA boxes. So here we've got a TATA box over on this side, TATA box which is leading to transcription of the, in this case, the TATA box over there is leading to transcription going in this direction. Now, hang on, the TATA box going in that direction, then TATA box going in this direction, the PL and PE promoters. So the TATA box here for this, and this is just the opposite strand, so that's why the TATA box is ATA, or AT, I should say, instead of TATA. So opposite strand here, again, large T-antigen binding. First thing that happens once this genome is released inside the nucleus is you get transcripts from the early messenger RNA because you don't have any of the large T-antigen around. As soon as you get more of the large T-antigen around, it's going to bind to sequences here which overlap with this AT box, which is going to block any more transcription which is going that way. And then you're going to get transcription from all of these, the late promoters. What's curious about this is they actually start at many different places, whereas the early promoter basically starts in just one place. How do you get transcription that happens? Again, the very first thing that happens, you get disassembly of your capsid in the nucleus. need to get transcription of these early genes. How does that happen? Happens due to cellular proteins binding to this sequence upstream of the P early promoter. And here it's labeled as being the enhancer, and this was the first enhancer of transcription ever to be found. So these are binding sites for cellular transcriptional regulators, and it serves as an enhancer because you can move this piece of DNA a long way away from this promoter. You can flip it over, you can move it downstream, it can be upstream, etc. So it's this, DNA sequence to which you have lots of cellular regulators that bind to it. And in fact, the first ever transcriptional regulators to be found in eukaryotic systems were found because they bound to sequences right here. In fact, called SP1, which is the very earliest transcriptional regulator to be found. So it's all about cellular proteins which are involved in the regulation of transcription from this P early promoter. And then what happens is later on in the process, you have the large T-antigen, which will block the transcription from the early promoters, and you end up with transcription from the late promoters. The other thing that we know about is that the origin of replication is right here in the same area, and we'll talk about replication a little bit later on. So now you're making these early transcripts. What do you get? So you end up with multiple different T-antigens, large, medium, and small, and in some cases even the tiny T-antigen. We're only really going to talk about the large T-antigen and then small T-antigen and middle T-antigens. We're not going to talk about the tiny T-antigen. Turns out this is only present in very few of the polyomavirus genomes and probably is not required. You can make deletions in these particular proteins and end up not with an issue. Now, what happens in terms of all of these T-antigens? They all have identical N-termini up to amino acid 79. So all of these proteins are identical. Then you have splicing, and that splicing just gives you different open reading frames. And so the C-termini of all of these proteins are different, but the N-terminal 80 amino acids is identical. So each of those are going to have exactly the same thing. What do these things do? Again, they're tumor antigens. What do we see that's happening with them? Basically, as a DNA virus replicating DNA, most of the time cells are not rapidly going through the cell cycle. So they're not undergoing replication. They're not going into S phase. They're not making all of the cellular proteins, including the DNA polymerases, all of the proteins that you need to make your individual nucleotides, etc. So basically, for these double-stranded DNA viruses to replicate, they need to convince the cell to go into the cell cycle. How does that happen? Well, it's basically the activity of these tumor antigens. The small T-antigen activates the MAP kinase cascade, and hopefully all of you are clear on MAP kinases. If not, you will be very soon in cell biology, I'm sure, but I always like to think of these as kinases of kinases of kinases of kinases of kinases of kinases. So there's the MAP kinase, which is basically the top of this cascade, phosphorylates the MAP kinase, et cetera, et cetera, et cetera. So you end up with all of these different kinases moving phosphates around, eventually leading to activation of transcription by some kind of transcriptional factors. This is activation protein 1, also known originally from SV40. And that binds to cyclin promoters, leads to expression of cyclins. Expression of cyclins, of course, leads you to be going through the cell cycle. Normally, this is regulated very much early on in these cascades by not just kinases putting on phosphates, is taking off the phosphates. And so the phosphatases, in particular protein phosphatase 2A, which is one of the very first phosphatases that's used in basically backing up this pathway, so inactivating the MAP kinase cascade. What the small T-antigen does is it binds to protein phosphatase 2A, so it can't dephosphorylate these substrates for the MAP kinase, So you end up with the phosphorylation cascade leading to expression of the cyclins. So that's one way that these T-antigens can stimulate the cells going into the cell cycle. The second way is through the middle T-antigen. Basically, what the middle T-antigen does is it pretends that it's a protein tyrosine kinase. So these are some typical protein tyrosine kinases. Sarc is just one of those. In fact, Sarc was originally found because it was in a retrovirus, one of these transforming RNA viruses, that RNA tumor viruses. But we know now that the cellular Sarc is a protein tyrosine kinase. Once you have an appropriate signal on the outside, which is usually for replication, these guys will dimerize, get tyrosine phosphorylation, then bind to a number of proteins that will lead to expression of things that will be important for going through the cell cycle and proliferation. And again, basically, all the middle T-antigen does is pretend that it's one of these phosphorylated protein tyrosine kinases. And then because of this phosphorylation, you have all the normal cellular proteins which would bind to that and be activated, which again lead to proliferation processes. So those two are relatively clear. Again, small T-antigen, which is blocking the phosphatases, allowing activation of the MAP kinase cascade, and this middle T-antigen, which leads to the activation of MAP kinases, but basically is pretending to be one of these receptor tyrosine kinases. Then
Wait, so is there no need for the middle T-antigen to dimerize with anything else?
It doesn't seem to have to be, no. It just seems to be autophosphorylating. So unlike, you're exactly right, normally these protein tyrosine kinases, they have cross-phosphorylation that takes place. Here it seems to be active as a monomer. So then we come to the large T-antigen. And this I like to think of as sort of the Swiss Army knife of replication and turning on all of the processes that you need to do to get a double-stranded DNA virus to be replicating. First thing to look at are these. So the proteins down here in the middle in brown, orange, and then all of the different things that it interacts with are all of these gray partners. And so each of the parts of this brownish, orangish genome here are going to interact with various different things. So the large T-antigen interacts with the RB protein. It has a nuclear localization signal. So again, just like we've talked about already, influenza, all of these things, all the genes are being transcribed in the nucleus. Those messenger RNAs are exported to the cytoplasm. They're then translated in the cytoplasm. They need to get re-imported back into the nucleus. Nuclear localization signal for large T-antigen was the very first nuclear localization signal to be characterized in any system. There's a J domain, which looks a lot like a DNA J protein, which is important for dealing with heat shock issues, p53 binding sites, p300 binding, which is a transcriptional coactivator. And then there's also, as you can see, lots of binding to things like DNA helicases, ATPases, DNA polymerase alpha, and the TATA binding protein, which is really important for transcriptional activation. So basically, large T-antigen binds to everything, more or less, at least everything you need for transcription, for replication, but also in terms of regulating the cell cycle. So large T-antigen is probably best known for its role in replication. You remember it binds to those sites near the origin of replication to block transcription from those early genes, but it also serves there as a helicase to open up the two strands of DNA in such a way that your DNA primase can come in, put down a primer, and then extend from the rest of the genome. So it is a classic helicase, binds to around DNA, and so the structure up here at the top, it's a hexameric ring, which basically the DNA goes down the middle of, and it will separate these two DNAs and really pump the DNA through this hole in the middle, and really by that process pulling it apart. Lots of ATP hydrolysis, On the last slide, I forgot to remind you, it also binds to RPA. What's RPA? Replication protein A. Really critical for replication. That was that last course, last term, or 2 terms ago. Completely wiped it out. Single-stranded binding protein. So this is the single-stranded binding protein that you need once you've pulled those 2 strands apart so they don't come back together. So large T-antigen binds to RPA, also binds to DNA polymerase alpha, which is doing what? Making lagging strands. Particularly, and topo1, which is important for dealing with supercoiling. The genome of SV40 is wrapped around nucleosomes, so it also has a supercoiling aspect. So let's look at this in a cartoon form. Here, the large T antigen is that hexameric ring together with the topoisomerase, which is dealing with supercoils as soon as you're pulling the 2 strands apart. And then RPA in the dark gray circles, again, this is a single-stranded binding protein. DNA polymerase alpha and the DNA primase, these are, again, cellular proteins which are involved in this process, make regular replication forks together with the pCNA sliding clamp, the clamp loader, and all of the replicative polymerases. In many textbooks, you will see when people talk about the eukaryotic replication origin complex, they'll have large T antigen written in here, which I think is really kind of funny because if they're not infected by one of these polyomaviruses, they don't have large T antigen in there. So it's probably the MCM proteins. But if you, again, look at a lot of textbooks, they'll still have large T antigen here because this is how people found out about origins of replication, what proteins were involved, et cetera. It was in studying SV40. Was there a question down here? Yeah. Okay. Okay, so we've replicated our genome. Now we need to make all of our structural proteins, so the late proteins. Large T antigen, as I already said, represses the early promoters. We've got cellular factors that are involved here. You also have differential splicing leading to VP2, VP3, and VP1. These, however, the splicing takes place before that AUG residue. So the N-termini of each of these proteins is different relative to each other. So this is a big difference then from all of these T-antigens, which all have the identical N-terminus relative to each other. And you can just see that. It's the thicker lines that are on here. Those are the protein coding regions. And so these are different starting positions for each of these different proteins. But you'll also see that here we've got overlapping open reading frames, a lot like what virus? Lambda, not really. 5X174, exactly. We've got lots of overlapping open reading frames. That also means that these things are highly susceptible to mutation because if you've got multiple overlapping open reading frames, one nucleotide change could be changing 3 different proteins potentially. So so although here one of the things that you'll notice is a lot of times the splice that takes place is actually then overlapping part of the small T antigen. It turns out you can replicate in the absence of the middle T antigen. The middle T antigen is actually not completely required. Small and large T do seem to be required, but middle T is not. Yeah?
So this picture is a little confusing to me. Are these different strands of DNA or just different reading frames or what's?
What are all of these funky lines? So the thick black line in the middle, this is your double-stranded DNA genome, so packaged with nucleosomes. Okay, then each of these thinner black lines are different spliced transcripts. So here, this one is going to be the transcript for the tiny T antigen, which we didn't talk about. But it's a transcript which has been spliced, and then the thick orange lines are now the coding sequence. So what's going to give you protein. And so those are where each of your protein coding sequences are. The dashed bits are where you have spliced out. So those are the introns. That make more sense? Yes, no, kind of, sort of.
So they're all the same length. It's just a matter
Well, they're going to be slightly different in length because, for instance, these guys have bigger splices than those guys.
Okay,
So, but
the initial template is all the same.
the initial template's exactly the same size, and so that primary transcript, which is made, is always going to be the same length. Then you get splicing that takes place.
Alright, so the splicing is what changes
The protein, so the coding sequence. Yeah. And all that splicing is taking place, of course, in the nucleus before that messenger RNA gets exported into the cytoplasm and then translated.
Is there any other viruses that do that?
W- That have alternative splicing like this?
Well, yeah, that the splicing is what determines what protein's going to be.
Well, HIV is a really nice example of that. And so that's important. All of the retroviruses, you have splicing to get your envelope proteins. So, yeah, splicing is really quite common for generating lots of different. Now that you have multiple different overlapping open reading frames, the only one that we've really talked about in not very much detail was HIV, because the TAT, the REV, the VPU, the VIF, all of these are overlapping open reading frames, but it's the differential splicing which gives you a different protein produced. Yeah?
The direction of the arrow shows which strand is being used, right?
That's correct, yes. So remember, it's the at here as opposed to the ta-ta going in that direction. So the bottom strand versus the top strand.
Makes a lot more sense.
Oh, and so instead of having right and left, you have early and late, right?
Yeah, it's just with the nomenclature here. You could call it right and left, but
So
yeah.
which one uses the?
So if you think about
which one uses which strand?
yeah, so of course these are all wrapped around each other, so it's really kind of immaterial which one's really the top and which one's really the bottom. But just in terms of the way this is written here, and just as easy to go around the other way, it's completely symmetric. But here, this would be bottom strand and that would be top strand. If you don't think about them being wrapped around each other. Yeah, but it's easier to talk about PL and PE because that just tells you a little bit about what those genes are that are being made. Okay, so assembly, capsids get put together in the nucleus. How you actually get the capsid, so it's a naked virus from the nucleus to the outside of the cell, is still kind of an open question, and nobody has a really good handle on exactly how that's happening. But we know, and you can see particles that have assembled in the nucleus, and you also see them on the outside, but the processes and how that's actually happening are still pretty open. So I wanted to spend a couple of minutes talking about some of the tumor viruses here. These polyomaviruses, again, usually don't cause tumors, but if you think about those T-antigens, they're tumor antigens, and they're really involved in stimulating the cell cycle. So if you really overexpress these tumor antigens, you can cause a cell, even in the absence of the rest of the virus, to go into uncontrolled proliferation. So it's those early proteins, again, the T-antigens that are important for this. Turns out you only get tumors in these so-called non-permissive cells, and we talked a little bit about permissive and susceptible cells earlier. So susceptible cells are those that have the appropriate receptor on the outside, and the permissive cells are those that can replicate the virus and such that it makes new virions and goes out. You only get tumors that form in non-permissive cells. So basically it seems that if there is a possibility to be making more viral genome and more of those virus particles, you're never going to form tumors. It's all just going to be making these virions. However, if you're in a cell type that's missing one of those cellular proteins that's critical for making those different either early promoters or late promoters, then you'll be making the early proteins but not making any of the late proteins. So those are going to build up. You're not going to have the regulation to shut down those early proteins. Then you can lead to tumorous development. Yeah?
So would that be from like a different cell type or the cell already being kind of screwed up?
So they're usually different cell types. So they're usually very different cell types. And it's 1 in about 100,000 of the non-proliferative cells that are infected, so they've got the binding site, the sialic acid, which of course is pretty common, that can be transformed. Few cases you see integration of these viruses into the genome, which is really kind of interesting, and how that happens is not entirely clear. Also very open area of research. So if you overexpress these tumor antigens in cells and they're not making virus, then you can get tumor formation. There's a little discussion in the textbook about Merkel cell polyomavirus, so how actually some of these polyomaviruses may be causing cancer in human cells. Turns out, and this is just a recent quote, basically it's not clear at all whether this is a causal or just a guilt by association process. And these polyomaviruses are really very common. You find them all over the place. So found in a number of these carcinomas, but not all of them, and it's really highly controversial. So I just wanted to mention that because, again, it mentions in the textbook this Merkel cell carcinoma, which I don't think is a causal. At least it's not been shown definitively to me that it's causal. So revisiting our polyomaviruses, they're, quote, tumor viruses. The structure is different. We've got early versus late transcription, and it's all about the T-antigens. So I did want to spend a little while talking about papillomaviruses. Turns out that the way these things work is pretty similar to the polyomaviruses. For a long time, they were classified as the same sets of viruses. These should look very similar to what we've already seen before, these hexamers packaged, but these really are tumor viruses. And so there are a number of human tumors, and particularly the human papillomavirus cervical cancer, which is caused by this particular kind of viral infection. There is one big difference that you have here, and that is that these viruses can replicate in differentiated cells, which normally would not be going through the cell cycle ever. And so they can convince those cells to start to replicate. And that's where we really end up with these tumor processes. So again, they're very similar to what we have in the polyomaviruses. Big difference is how they're being regulated in terms of their transcription and also what some of the early proteins are doing. Most papillomaviruses, human papillomaviruses, lead to warts, which is not particularly attractive, but also not terribly concerning. In terms of disease, they are clearly infectious. I think the last count, there were 150 different human papillomaviruses out there, so they're huge numbers of them, mostly okay, except, of course, for cervical cancer, which is this squamous cell carcinoma. How do they get into cells? It's a little different than the case for the polyomaviruses. These guys bind to basal epithelia. So this is the epithelial cells that are skin cells. They're all the epithelia in our guts, all of the other places. Heparin sulfate proteoglycans is the glycoproteins on the outside of the cells. Integrins, they come into the cell with normal endocytosis, so not the caveolins, and then express these early genes. Again, this seems to be happening all in the nucleus. You get the particle coming in, that particle then gets transported to the nucleus. It's opened up in the nucleus. You have also double-stranded DNA packaged in nucleosomal structures, but instead of having divergent promoters, you have overlapping promoters. And that's just shown here. This is really a circular genome that's just shown here to be, I think, a little bit easier to look at. Got our early proteins and our late proteins. Also has a regulatory sequence, locus control region, which is present here. This is very similar to what you have in the polyomaviruses. Lots of cellular proteins bind to this control region. Again, these are naked DNA viruses. They don't bring any of these proteins with them, so you have to get transcription of these proteins at that process. So early and late promoters, the big difference here is these guys overlap with each other. So instead of going in opposite directions, they're overlapping. The early promoters make the early proteins, particularly E6 and E7, which turn out to be the most important for tumor formation. And then the late genes, which also include, confusingly, the rest of the early proteins other than E6 and E7. So E1 and E2 are going to be the ones we're going to spend most of the time talking about. L1 and L2 are basically identical to the L1, L2/3 that you have in the polyomavirus in terms of how they're being made. So those are not that critical. What's interesting is that the late expression and the expression from this promoter depends on also cellular proteins, but these are cellular proteins that are expressed in, and so getting different than what we talked about before for permissive versus non-permissive cells. These are now expressed in different states of differentiation. So in normal epithelia, you have down at the bottom your basal epithelia, which is where the virus is doing its infection process, and then the differentiation process where you end up with all these dead cells at the top. So skin is replicating all the time, but it's replicating due to these stem cells, and then as they differentiate, they get up here to the top. What's really different in terms of the HPV-infected cells is they maintain their nucleus and maintain this replicating form all the way up to the surface of the epithelium. And so that's where you have the big difference. And then, of course, this is what's leading to your tumors right here, is this differentiation up the top, and also wart formation, because these are growing cells that are forming on skin, for instance. So that expression of those late genes is dependent on the proteins that normally would be causing nuclear degradation, and these cells that are no longer nucleate here have been co-opted in terms of being used to make the viral proteins. So let's look a little bit at these proteins. Again, it's just the table. Important here are going to be E6 and E7 that we talk about in terms of the tumor proteins, and E1 and E2. So E1 serves very similarly to the large T-antigen. It's a DNA helicase, separates the two strands, allows interaction with all of the cellular replication machinery. E2 is a specific binding protein that helps E1 to bind to the promoter, but also serves as a transcriptional regulator. And so these are those two proteins, and it seems to be these two proteins that are basically serving the same role as the large T-antigen. So E1 and E2 in this process. E6 and E7 are the ones that are bad, i.e. the ones which are leading to tumor formation. And it's all about interactions with RB and P53. So hopefully you've all heard about RB and P53, the classic tumor suppressor proteins, mutated in large proportions, and I forget exactly what the numbers are, of solid tumors, and also in a lot of the non-solid tumors as well. How many people are taking this cancer biology class? Any of you here taking cancer biology? Okay, yeah. So you can probably tell me more about this than I can. So the PRB is really a regulator of the cell cycle. And so down here at the bottom is the normal process of what happens with RB. Normally, RB is associated with E2F. When E2F, who knows what E2F stands for? These are papillomavirus E2 regulated factors. So these are transcription factors which are normally regulated by the E2 protein from papillomaviruses. So they're called E2Fs because we originally discovered them in studying papillomavirus. It turns out they're in all cells. It's not, these are normal cellular proteins which are there. So normally, RB is associated with these E2F. In the normal cell cycle, cyclin and CDK is going to phosphorylate the RB that releases these E2 factors, which go ahead and transcribe lots of cell cycle genes. So this is the normal process. P53 normally is going to activate P21 in things are going bad, which is going to block the activity of this, so RB stays associated with E2F. You don't go into the cell cycle. What happens with E7 is E7 binds to RB such that these E2 factors are released in the absence of going through the cell cycle, the normal cell cycle processes in cyclin and CDKs. And so basically what happens is you stimulate going through the cell cycle even when you shouldn't be. And so this is the interaction with E7 together with RB. As we'll see in just a second, E6 also interacts with P53. The SV40 large T-antigen interacts with RB and P53 as well. And so that's probably when you overexpress the large T-antigen leading to cancerous development, how that's happening. The E6 protein basically takes out P53. So P53, I mean, is the guardian of the genome. So when there's DNA damage, it blocks the cell cycle until you have a chance to go through the rest of it. If stuff is really bad, leads to apoptosis. But if you get rid of P53, you can't block the cell cycle when you have DNA damage or virus infection, and you can't go to apoptosis. So getting rid of P53, getting rid of RB, so it's sort of the, these are oncogenes because they are blocking all of the tumor suppressor proteins. So it's a blocking of that. So here, again, looked at P53 and P21 before. Normally, P21 is going to block RB activation, but no P53, no P21. These transformed cells that you've expressed E6 and E7 in, really similar to those polyoma cells. Turns out they're also not competent for virus replication, so these are non-permissive cells. And again, it's this, normally you'll have regulation, and we didn't talk about it, but those E4 and E5 that we didn't talk about are important for regulating E6 and E7. But if you're not making E4 and E5, then you're not going to be regulating E6 and E7. So again, it's very similar to what's happening there. So just to finish up, cervical cancer, E6 and E7, this is how you get these cells. HeLa cells are cervical cancer from Henrietta Lacks. Wonderful book on that. I'm not going to sign it for the class, but really interesting reading. These turn out to then integrate the papillomavirus into the genome, but it's just E6 and E7 that you need. You can take a cell, overexpress E6 and E7 in them, that will become a tumor. That's partly why people are screening for E6 and E7. And then my last slide is vaccinate, vaccinate, vaccinate. These are really interesting vaccines because they're recombinant vaccines where they're virus-like particles that just have the L1 protein, so basically that capsid protein. And so it turns out they're unfortunately rather expensive to make, but they work really well. But you have to immunize before infection, and these, at least the cervical cancer, papillomaviruses are sexually transmitted disease, so before sexual activity, this immunization should be done. There's all kinds of arguments about, well, if we get our kids immunized, then they're going to be much more sexually active because they... Okay. We'll just leave it at that. Have a good weekend, and we'll see you on Monday for review.